Thank you very much for introduction, for the invitation. It's a really great pleasure to be here and tell you about the research that my group have been doing since 2020 when I started at Delaware. So the main topic of my presentation is ion channels. But before that, I'll use maybe 5 slides to just introduce you to the other projects we have been working on in the past. So this is my ion channel slide, but you hear more about ion channels very shortly. So another direction that we go in is we're really interested in understanding liquid-liquid phase separation in biology, or specifically in intrinsically disordered proteins. So this is a very quick simulation of a big protein that has about 100 disordered chains interacting together, forming condensates. So these condensates are actually very important in cells. They work. It's essentially a cell mechanism to bring components together to react, because, as you can imagine, the intracellular environment is really crowded and very dynamic and very messy. But it's actually very interesting. But the chemical reactions occurring there, they're very precise, right? The question is, how does the cell achieve that precision? Very remarkable spatial and temporal control over what's going on inside. And one of the mechanisms is to form these droplets. So some biological systems like proteins and RNAs come together, they increase local concentration to enable certain chemistry. And we're really interested in understanding the mechanism, how this happens. And in one of the simulations, we look at the very important protein that's implicated in some neurodegenerative diseases. We found out that what brings them together, what brings the protein chains together, is the interaction of the side chains of a very interesting and very different nature. So we have pi-pi interactions, we have cation, we have electrostatic interaction. We also have, here is a cation pi interaction. And all these interactions together with hydrogen bonding held this condensate together. And what's interesting is that when the solvent, in this case water, gets inside of this condensate, its dynamics slows down. So this shows the distribution of characteristic time scale, the dynamics of water molecules near the protein when it's in dispersed phase, when it's all separated. And compared to the condensate, and as you can see, the dynamics becomes 5 times slower. So this is very interesting, another aspect of a liquid-liquid phase separation. Another continuing on the biological topic is we recently published this very nice PNAS paper in which we collaborated with NMR and 2-dimensional infrared spectroscopists at the University of Wisconsin, and we were able to determine the structure of human amylin oligomer. So human amylin is a short peptide, about 38 residues, and the aggregates of that peptide were found in pretty much every patient diagnosed with type 2 diabetes. So essentially these things, normally they disordered, but under certain conditions they come together, aggregate in the rib cell membrane, and that's one of the very simple explanation of hypothetical mechanism of formation of type 2 diabetes. So the initial structures are known, the final structures have been very well characterized. What has not been determined is this intermediate step, which is believed to be the most toxic, and it was very hard to determine this intermediate step, the oligomers, going from monomers to fibrils. The oligomers were really difficult to determine because they're transient. They form and they grow into larger fibril aggregates on a time scale of hours, which is very difficult for NMR to capture. So we have to engineer a mutation that stops this process, get those oligomers do an NMR, and get some details about the structure, some distance constraints of the structure, and then we use an extensive set of molecular dynamics simulations to produce a structure that's consistent with every experiment that has been done. So what you're looking at is a dimer, is the first ever published structure of the dimer of HIPP, which is a intermediate on the way to fibrils. It's very related to type 2 diabetes. Now, we also do a pure, essentially, theory work. We're really interested in understanding the multidimensional spectroscopy. This is very different from what you just heard about biological stuff. Here we have a sample. It could be a molecule in solution, of course, or could be a condensed phase system, or it could be solid state system. So what's happening in these experiments is this system interacts with a sequence of laser pulses which trigger transition between electronic states, and then the dynamics of those excited electronic states is measured eventually, and it produces a plot which is a function of excitation and detection frequency, from which you can learn about the excited states themselves, the coupling between the states, the dynamics between the states. And we recently have been interested in something that's called action detection spectroscopy. I'm not going to go into details of that. It's in this upcoming paper. Another new direction of theory development is terahertz IR spectroscopy. That's a very unique way of actually determining the coupling between low frequency and high frequency motions or vibrations. So this is the experimental spectra of water. It shows their signal between low frequency motions of water, vibrations, translations, and a high frequency OH stretch. So it means they're coupled. You can think about it if you have a water molecule, OH stretch, all right, motion, and it's hydrogen bonded to other water molecules. So this collective vibration, collective motion, which is a low frequency motion, is coupled to high frequency OH stretch. So we're developing theory for these types of spectroscopy. These are fairly recently developed experimental techniques. We're also interested in understanding the energy transfer in molecular aggregates. So that's the specific system of choice that we're working on fairly extensively. So it has several porphyrin rings, porphyrins connected into a ring-like structure with a butadiene. So this is a carbon-carbon triple bond linker here. And when the system is initially excited, what linker does, it directs the energy to these two porphyrin sites. And so this process occurs on two time scales. One is ultrafast. It's when the initial delocalization of the excitation happens. But then on the time scale of tens of picoseconds, we have the energy transfer to these two sites. Why this is interesting is because these time scales exactly mimic natural light harvesting complexes, which harvest light and transfer the excitation to a photosynthesis. So that's the initial step of the photosynthesis that is very efficient in many bacteria and plants. And so we do it by simulating two-dimensional infrared spectra, two-dimensional electronic spectra. So we're looking at the cross peaks that are signatures of that energy transfer. We've also published quite extensively on our new method for doing non-adiabatic dynamics without solving a time-dependent Schrödinger or Liouville equation. Our approach is based on using machine learning. So essentially we train a machine learning model on a short time dynamics, so you only allowed to observe the quantum evolution of your system for a very short period of time, and then you use that information to train a model that predicts the dynamics to arbitrary long times. So obviously that saves you quite a lot of computational effort in doing this explicitly solving time-dependent problem. So we're also interested in water, understanding its structure through vibrational spectroscopy. So we published a few papers, and we're going to focus on that. The most recent and really exciting work is, which also continues, but we already published a few papers on that, and it's we're trying to understand how the dynamics of the monolayer deposited on an electrode can be changed by changing the voltage. So that's the system we're looking at. It has this gold electrode, a molecule with a nitrile group. The tip of the molecule is CN, carbon triple bond nitrogen, and these are ions in water. And what we do is, both in simulations and our experimental collaborators do the same. They change the voltage here, they change the potential, and they study the monolayer dynamics. So it's a tilting of this molecule as a function of voltage. And what we discovered is a remarkable slowdown in dynamics when you add salts, electrolytes. So for just water, if you have no ions, the dynamics is relatively fast. It is voltage sensitive, but it's relatively fast. But if you add ions, it doesn't matter which ones you add. The positive or negative voltage brings different types of ions to the surface. The positive voltage, we have more chlorides, negatively charged ions coming down. For negative voltages, we have positively charged ions coming down. And in all cases, we found a substantial 6 to 8 times slowdown of the dynamics. And if you're curious why this is happening, partially the answer is in this paper, or a better answer and more complete answer is in the paper that we're currently working on. All right, now I'm going to talk about ion channels. And before I start talking about ion channels, I introduce the key people. So this is my group in this summer. And the work I'm going to be talking about was done by my postdoc Kyle and my 2 graduate students here. And it was a collaboration with the experimental ultrafast spectroscopy group led by Martin Zani at the University of Wisconsin and the structural biologist Francis Valivetil from Oregon Health and Science University. All right, now what are the ion channels? So the ion channels are transmembrane proteins that span the cell membrane, and they facilitate the motion of the ions in and out of the cell, right? Because this is very interesting design by nature, how ions can overcome a essentially non-polar environment of the biological membrane, right? They go from polar intracellular to polar extracellular, but they have to cross the membrane, and the ions are charged. So which means that this presents a problem for them to go through. And the design that was widely adopted is through creating these pore-like proteins. So these are proteins with a pore through which ions move. And this is very important because they actually participate in formation of the action potential. So this is a neuron and the signal going down the axon. By changing reversing the polarity of the membrane, here we have essentially a propagation of electric signal, right? And it all happens through this process, and it involves sodium ions moving in and potassium ions moving out, and it's all controlled by a very orchestrated motion of or function of ion channels. Now, ion channels are targets for neurotoxins. They're also associated with a lot of nasty disease. They call it channelopathies. And also a relatively large fraction of pharmaceutical markets, so that's all the endowments of Ivy League schools combined together will be smaller than this number. That's how much of the pharmaceutical market are drugs that target ion channels. So ion channels are very important, right? And they are actually implicated, as I will say right now, they're actually expressed in every kingdom of life, every tissue, your brain, your heart, it's everywhere. Different kinds of ion channels, of course, but they are everywhere. And in our work, we're specifically looking at potassium channels because potassium is the most abundant cation in cytoplasm, and there is a huge variety of potassium channels that are responsible for very key function for very key functions. And what's very interesting about them is they have a very selective conduction. So this is the selectivity, right? And it's very interesting because sodium is actually smaller in size, right? You would think if a bigger thing feeds, then a smaller thing would surely go through, but that's not the case. And the conduction also occurs with essentially a diffusion limited rate. So the ions go through the channel as fast as they come up to the channel. Yet it's very selective. So how does that happen? Well, hopefully I'll explain, well, nobody really knows, but hopefully I'll explain you our approach to answer that question. And so this is a MD movie, ions moving up through the channel. All right, so there's a large family of potassium channels, and the channels can be activated using different mechanisms. Some of them can be activated by ligand binding, some of them can be activated by changing the pH. there are many different ways to activate channel. And specifically, our work focused on the KcsA channel, which has become a quintessential potassium channel because it's a relatively small protein. That means it's easier to synthesize, characterize in the lab, and do experiments with. It is bacterial channel and it's pH gated, but it is functionally and structurally very similar to eukaryotic potassium channels, the channels that we have, and it has all the characteristics that I mentioned before. So I have a few renders of the structure. This is a top-down view. This is a side view. What you really need to learn is that this is a tetramer, so there are 4 identical units come together and form the channel, and these are the ions inside. This is a side view of only 2 of the units. Clearly there is this part in the middle that's called pore loop or P loop that holds all the ions. And if we look closely at this structural unit that's shown here, this is the actual X-ray structure of it, obtained in 1998, for which McKinnon received the Nobel Prize, I think 5 years after it. So this was the first structure. This is the first structure of ion channel that was resolved in experiment, and it clearly shows that ions occupy the central site in these specific spots. All right. And this is the ion that actually comes to the channel. So the ions move from bottom to the top. So here they come fully hydrated with water, and then they dehydrate and go through the channel and get out. Now, what are these red things here, red groups here? Those are the carbonyls. So this is a more schematic view of the central unit. It's called selectivity filter. This is where all the magic happens, well, where all the selectivity, as people believe, happens. And ions here are shown by these green spheres, and the carbonyls from the protein backbone come together to form this site. As you can see, every potassium ion is held by 8 carbonyls, so it's coordinated by 8 carbonyls in every of these sites. There are 4 sites in total, labeled S1 through S4. This is a very narrow part. It's actually only 4.5 angstroms wide, so you can only have 1 ion, maybe you can only have 1 water, but you cannot have 2 ions sitting in the same spot. So what ions do, they come up to the channel here, they enter the channel, and then just go up and then leave. This is what happens. Now, there are, of course, many questions in structural biology of how this happens. So we don't know the exact transport mechanism, which I'll address in a second. We don't fully understand how it can be both fast and selective. So the molecular basis of selectivity itself is not known, and we also don't fully understand how channel inactivates in various conditions. And there's always a question of how good MD simulations are, and every of these questions is essentially a decades-long debate in the community. And the first, I'm going to introduce the first question. That's the question of ion transport. It has been proposed that ions move through the channel being separated by water molecules. It has been supported by some experiments, for example, in streaming potential measurements. Those measurements show that water actually comes out of the channel. So it's not just ions that go through, but water is there too. And this mechanism has come to be known as soft knock. Sometimes it's called water-mediated knock-on, but I'm going to refer to it as a soft knock for short. This was the one that's originally proposed based on the X-ray structure, so in the previous slide, and it's now essentially a textbook mechanism. However, in recent years, these mechanisms has been challenged by MD simulation, some reinterpretation of X-ray data, and some NMR experiments that show that there is no water in the selectivity field. So you just which of this mechanism dominates. The very early view was, as I mentioned in the previous slide, that soft knock dominates. Some numbers from the papers that goes back as essentially to the time that the X-ray structure was revealed, essentially shows that there are very low activation barriers for soft knock, so it all makes sense. And if you replace sodium or potassium with sodium, then free energy goes up, so that explains selectivity. However, if simulations are done in non-equilibrium, in this case meaning in the presence of transmembrane potential, then the simulations typically only favor hard knock configuration. Yes, and I need a mic.
On the previous slide, you said they do X-ray crystallography. So my maybe I'm wrong, but my understanding is that if you want to do X-ray crystallography, you need to be at relatively low temperatures and freeze the sample, right? So how do you know that how can you observe water going through or not if the water is frozen?
You don't observe water going through in X-ray. I think here I mentioned it's a streaming, but this is a different kind of measurement. This is not an X-ray.
Oh, I see.
So this is what you see in X-ray. This is from the structure, and so it shows what it is basically a crystal, right? It's averaged over many channels, and it basically tells you that ions can be at any of this site. But you're right. This is a low temperature crystal, has nothing to do with real biological system. Very little. Okay. Right. So some simulations, especially those that are done under transmembrane potential, favors hard knock. So this plot shows the current as a function of voltage, and this is a hard knock. This is soft knock. Essentially, the simulations show that there is no current, no conductance for soft knock configuration. So there's this controversy in the field, soft knock versus hard knock, which hasn't been addressed experimentally and in simulations. The approach that we took with my colleagues is the following. As I mentioned already, the interior of the selectivity filter is composed of these carbonyls that point inside. And in vibrational spectroscopy community, carbonyls known as a very good markers of the local structure. What I mean by this is the following. This is a very famous plot showing the CO stretching frequency, carbonyl stretching frequency as a function of electric field. So it's a linear, it could be interpreted in terms of vibrational Stark effect, which tells you that the frequency shift is proportional to electric field. It's a little more complicated than just Stark effect, but I'm not going to go into those details. What we need to see from this plot is that the stronger the field, the larger the shift. All right, and it's a pretty big difference going from nonpolar solvents to water in terms of a frequency shift. So this essentially means that if we can somehow take a real ion channel in its environment, maybe in the membrane, and measure somehow the vibrational frequencies of these carbonyls, we should be able to tell the difference between ions and water sitting here. At least we should be able to tell the difference between two ions here or ion and water, right? Because two ions should give a large field, much larger than ion and water. And a quick simulation does indeed support this. If you just put an ion in the center of the binding side, then you get this massive frequency shift. If you just put water, it depends how you exactly put water in there, the frequency shifts are much smaller. The question is, okay, if we can measure this, how do we know that we're measuring these carbonyls, because protein has thousands of those? And the answer is that we use isotope labels. So instead of, naturally it's carbon-12, oxygen-16, if we replace them with carbon-13, oxygen-18, through a chemical synthesis or semi-synthesis and expression, we can change this carbonyl into 1318, which changes the mass, which changes the frequency, right? This substitution changes the frequency by almost 70 wave numbers, meaning that we can isolate these carbonyls out of the signal from everything else. It's a big change, 70 wave numbers, you see that. Turns out it's possible to prepare protein with isotope labels exactly where you want to put them, right? Because you synthesize this protein, you buy an isotopically labeled residue, and when the time is right, when you build the sequence, the next residue you provide in your in your synthetic tool is isotope labeled residue, so it gets incorporated exactly where you want it. And then, of course, you have to express protein and all that, but I'm not talking about that. That's an experimental detail. All you need to know here is that it's possible to precisely put the isotope label inside of this selectivity filter, and it shifts the vibrational frequency enough so you can resolve the signal that's coming from exactly that side. All right, the technique that's used to measure vibrations is a two-dimensional infrared spectroscopy. So I'll spend a little bit time explaining what it is. So it's essentially that helps. Sometimes it depends on the audience. It helps. It's an optical analog of 2D NMR, which is way more known, especially in the biophysics community. So what it does is probes molecular vibrations. NMR probes spins, nuclear spins. 2D IR probes molecular vibrations. So if you have a molecule, very simple molecule like this, it vibrates, and then you subject molecule to multiple interactions with the laser field, what happens is that you trigger this transition in electronic states, in vibrational states in this case. So this is ground state. The first pulse can excite to first excited state, and then the second pulse can either further excite through excited state absorption, or it can go stimulated emission, or it can hit a different molecule and go and trigger 0 to 1 transition. All right, so all these laser pulses do all this, basically climb up and down the vibrational ladder, and the spectrum that you measure looks like this. The first time is the Fourier transform of the first frequency, omega 1, is the Fourier transform of the delay of coherence time here, and omega 3 is you could detect from by the detector, or you can do Fourier transform of detection time here. And so the spectrum for a single vibration looks like this. It has two peaks. The red one, it's right on the diagonal. Excitation is the same as detection frequency, and that corresponds to 0 to 1 transition, or 1 to 0, same frequency. Another peak that always accompanies the red one is the blue one, that corresponds to 1 to 2, which is shifted because of anharmonicity, right? This is anharmonic, vibrations are anharmonic, so you get peaks like this. If you have two vibrations, each one of them shows up at the corresponding frequency, but you can also have cross peaks, and the cross peaks tells you about the coupling between the two vibrations. And the time scale of these experiments are femto to picosecond, so they report on picosecond dynamics. In the protein case, it really basically means that we measure static snapshots, because proteins don't move on femto to picosecond time scale to a sub-picosecond time scale. But the solvent near protein can move, and I'll come back to this later. And my group does simulations. So to simulate these experiments, we need to simulate a third-order optical response of the system. So essentially, this is a microscopic polarization, the quantity of interest, and you can think about it as a convolution of electric fields and something that's called optical response function. And the optical response function has all the information you need to know about the molecule, frequencies, dipole moments, all that. And to simulate this function, we need to consider all the different pathways we can take through the vibrational states. I'm not going to go into details of that, but you can grab a very famous book, and it explains all of that. And that's the quantity we need to simulate. And what you need to know to simulate this is the dipole moments, which are shown by m and mu here. There's a difference between m and mu. I'm not going to go into that. But so you need to know the transition dipole moments of the system, and you need to be able to solve the dynamics of the system in between. Right, so you hit the molecule with a laser pulse, you cause one of these transitions, and then the molecule just evolves in time until the second laser pulse comes in and hits the molecule. So we do it 4 times, and then we do 2 Fourier transforms, and we get 2-dimensional plot. All right, now how do we incorporate molecular details in that simulation? And in case of proteins, we oftentimes look at CO stretch, as I already mentioned. It's called amide 1 mode. That's the name of it. And we use an approach that's called mixed quantum classical approach. Essentially, it uses molecular dynamics simulations to sample relevant configurations of the system. But as I said in the previous slide, you need to know vibrational frequencies and transition dipole moments. And a specific approach that has been widely adopted by the community is based on vibrational frequency maps. So you have your MD simulations, you have lots of snapshots, and for every snapshot you need to calculate vibrational frequency, which for amide one has been given in terms of 4 components. This one is electrostatic, so it gives the dependence of vibrational frequency on the local environment. So these are electric fields. So if ion comes in, it changes electric field. This is electric field on carbon, electric field on nitrogen. So ions and water in the local environment change these electric fields, and they change the frequency, right? Frequencies also depend on the nearest neighbor proteins through dihedral angles. That's not the focus of this work, but it's there because that's the part of the equation. And this is frequency shift for isotope labels. But this is important. This is how frequency depends on the electric fields, right? And as I said in the beginning, electric fields are changing as you change the local environment. And this is the definition of the transition dipole moment we use from the paper. So this is how it's oriented along the CO. It's almost parallel to CO stretch. All right, and going back to the first question, do we have water in the selectivity filter or not? So we've done the experiments exactly as I described. So the synthesized protein, we synthesized, well, not we, but our collaborators in Oregon synthesized the protein, shipped it to Wisconsin, where the 2DIR spectra was taken. This is an experimental spectra. As I already walked you through this should not be a new thing for you. We have 2 peaks, red and blue. Right, and this is the frequency around 1590. This is the vibrational frequency of CO straight shifted due to isotope labeling. And so this is our simulation for soft knock. So you see water and ions are there, and this is our simulation for hard knock. No water here, and because many hard knock configurations have been proposed in the literature, we had to simulate basically all of them. But none of them really gives anything close to experiment. This is the best you can get. Looks very different. However, soft knocks looks very good, right? And in this paper, we essentially concluded that in the conditions of the experiment, soft knock must dominate because there's very good agreement for soft knock compared to hard knock. And the conditions of the experiment are, of course, important. Those were equilibrium conditions, and the channel was in closed and conductive state, meaning that it was in this state, or on this state. The entrance of the channel was closed, but ions could diffuse in and out. So that's the condition of the state. So the selectivity filter, we believe, was fully functional, so ions could come in and out, but the channel itself was closed. All right, so the next project that was kind of a spin-off of this project is that we looked at the water inside of the selectivity filter. Now that we convinced ourselves that there is water, we asked the question, what is water doing inside the selectivity filter? Is it moving, not moving, hydrogen bonding, not hydrogen bonding? What is it it's doing? And for that, there's an extension of 2DIR experiments that can report on local dynamics. And in experiments, we're focusing now on the waiting time. Remember, the 2D plot is a Fourier transform of these, gives you one axis, then another axis gives you the Fourier transform of these, which means that it's all really parametric with this waiting time. So it could be any time you want, and you can control this in experiments. Right? If you set this to 0, let's think about that case. So the first 2 pulses arrive and hit the sample, so you get the first frequency. And then you immediately hit the sample with the 3rd and 4th pulse, and because you did not wait anything, right, any time, so the other 2 pulses detect exactly the same frequency, right, because there's just no time between. And so it's the waiting time 0 experiment. But now, if you set a delay between the first two and the last two pulses, what happens is the first two pulses come hit the molecule, and they essentially detect or probe or record one frequency. But then you wait, and during that waiting time, the local solvent can move, right? Because you wait, and your waiting time are picoseconds, and we know water moves on picosecond time scale. So essentially, you wait, you allow solvent to move around, change the frequency, because solvent changes the frequency through that equation, right? Frequency is a function of electric field. As solvent moves, electric field changes, frequency changes as well. And so the last two pulses will now detect a different frequency, right? Because now there's a gap between the first two and the last two. And you can play with the waiting time, all right? So you can increase the waiting time, and the spectrum goes from this is waiting time 0, to this. This shows that you have correlation between excitation and detection, exactly how I explain it, right? Because you don't wait, so you detect the same frequency as you probe. But then as you wait longer, those carbonyls experience all the different environments due to solvent, which causes decorrelation between excitation and detection frequency, and the line shape is basically round. Now, these are idealized line shapes. This is a real experiment for the dipeptide done by Takmakov group. This is essentially 0 waiting time, so this looks somewhat like this. But then when you wait till 5 picosecond, this is much more round now, right? So again, these are idealized. This is what you see in real experiment, but I hope you see this, that the line shape going from stretched to now more round, right? And if you plot a function that shows how fast, basically the slope of this, it looks like this. So the slope of this is the line that you see here, right? It's changing, and if you plot the slope of it, it's called central line slope, and this is a central line. It decays like this, and then you can feed mono by exponential function to extract the time, and in this case, in this experiment, the time is 1.2 picosecond, which is exactly the water dynamics near those carbonyls. So this is how you measure local solvent dynamics using 2D IR spectroscopy. Okay, so these...
Sorry, yeah, these were not labeled, were they? These are like solvent, like a bulk solvent, basically.
Yeah, this is bulk. This is just this dipeptide in water. Yeah. Okay, we apply same idea to carbonyl residues in the selectivity filter, and so there are many more line shapes of different waiting times that you can find in this paper. So I'm showing just 500 femtosecond and 2 picosecond. All you hopefully see is that there's no change, right? This is experiments, simulations reproduce the experiments. There's no change in the central line, and this is, that the function that was decaying for the dipeptide in the previous case. Now, if you plot the same function for our case, for these residues in the selectivity filter, it doesn't decay. Means there's no picosecond dynamics. And then you say, okay, that's obvious because ions move on nanosecond time scale, right? Why would I see a picosecond dynamics? Well, we were hoping that we'll see water because water moves on picosecond time scales, but we don't. Now, the question is why? And here's the answer. It turns out that so this table gives you the frequency shifts due to various components. So this, for example, the protein, electrostatic environment, and structural changes cause a frequency shift of about, between 50 and 80 wave numbers, but water causes a very small frequency shift. And really the major contribution here is ions. Ions shift the frequency by 100 wave numbers, which goes all the way back to one of my first slides where I show a very simple calculation essentially reproducing this. So yeah, because ions dominate the line shapes, a very little frequency shift from water, you just don't see it. So that's the answer. But it was still very interesting for us. So basically experimentally we cannot do that, right? We do not see water moving. But in simulations you can, in simulations you can certainly see how much water moves, and a quantity relevant to experiments that simulations can do is called the frequency correlation function. That's exactly what I explained 2 slides back. It shows how fast the frequency decorrelates with itself over time. Typically, it looks like this, right? If it's normalized, it starts from 1. And there's a big initial drop that's caused by a local rotation and translation. And then there's a slower decay on about a few picosecond time scale. If you have hydrogen bonding, it contributes to it. And a longer time scale, the diffusion would contribute to it as well. In our case, ions, this longer time scales due to ions would be nanoseconds. All right, this is just general. This is not, this is a very nice illustration, but this is not the result. This is our result. And it shows, if we just focus for a second on the full line shape, that's, I think that's purple. As you can see, we do see this initial drop, but then it doesn't decay, or decay very slowly, which is exactly what you see in experiments. Nothing really changes on a few picosecond time scale. But what you can do in simulations that you cannot in experiments is you can break down this correlation function into different components. You can ask the question, how much of this frequency change or frequency correlation is due to ions, how much of it is due to protein, and how much of it is due to water. And that's what you see here. Ions are shown in blue. It's a straight line after initial decay. So initial decay means ion just moves a little bit within the binding site. But then it doesn't go anywhere else. So it just stays there, and that's why this line is just flat. It's not decaying. Same for protein. There is no structural protein motion on a few picosecond time scale. But if you look at water, red, so water does decay, right? It's not very fast, but it's much faster than protein and ions, right? Hopefully you see the red line decaying a bit faster than the blue and the orange, right? So water does move, so we were interested to see what exactly water is doing there, and you can just come up with the different ways water can arrange itself in the selectivity filter, right? We know that water can hydrogen bond to carbonyls, right? To 2, to 1, or it can just kind of stay in the center of the binding site here. So then we'll look at all of these simulations that we have. That's about 100 nanosecond simulations that we've done. For these, we were surprised to see there's only 3 types of water, right? Out of 8 types here that are possible, right? We saw only 3. And one type of water is when it's hydrogen bonded to adjacent carbonyls. That's orange. The blue type is when the water is hydrogen bonded to next neighbors. And the green type is when when the water molecule is not hydrogen bonded to the carbonyls and it's just kind of being coordinated by an ion, but essentially sitting in the middle of the binding site. We calculate a few important quantities to describe these water molecules. And the first one is hydrogen bond rotational correlation function. Compared to liquid, so these colors match these colors. As you can see, we have a slowly decaying correlation function. This is OH bond, right? So this is a correlation function of OH vector with itself. So it's decaying, but it's decaying much slower than liquid, for example, and the other 2 types of water. On the other hand, we have red, or have green, which decays so fast, right? It's much faster, and it's because it's just spinning basically in the middle of the binding site. This is hydrogen bonding correlation function. All the hydrogen bonds are very short-lived compared to liquid. Magenta is liquid water. Hydrogen bonding inside of the binding sites is very, very short. So it's very interesting, right? So we see the different types of water. One is slower than liquid, one is faster than liquid. And now this doesn't decay to 0 because this is known in the water community as wobbling in a cone. So this water molecule essentially wobbles like this. So it breaks the hydrogen bond, reforms hydrogen bond, but it never leaves these 2 carbonyls, at least on a 10 or longer picosecond time scales. It just sits there. It's known in the water community in different contexts. We could also estimate how much ions move. All right, this is a distribution of frequency shifts. We know it peaks about 110 wave numbers, but it's actually fairly broad. And you can use this to calculate how much ions can move up and down, because this frequency shift is given by the electric field, and the electric field is ultimately given by the distance between the ion and the carbonyl. So you can do the math, and we did it, and we found out that ions can actually move quite substantial. They move up and down about 30 to 40% of the distance to the carbonyls. And then we learned that, it was previously observed in empty simulations, but here we came in from a vibrational frequency sort of independent point of view, and we were able to reproduce the magnitude of the ion motion observed in pure MD before. All right, so that's my conclusion for this part, and I should probably end here. There's a third part of my talk. It's about selectivity. Here the focus of this part of the work is that we design an experiment, a 2D experiment that will tell you the difference between sodium and potassium binding in the selectivity filter. Turns out that they bind differently. This is the potassium binding in the middle of the cavity, and the sodium binds in plane, which is very different. It has been observed in X-ray, so this is not a new result, but our simulations show that you can use 2D-IR spectroscopy to detect the different types of binding, because the spectra for potassium and sodium are very different. Well, there's a significant shift, which you can definitely resolve in experiments. So hopefully, so we propose these experiments to our colleagues. Hopefully, experiments like this will be able to give us new insight into selectivity in ion channels. So another new direction for us is now voltage-dependent conformational changes in the protein. So now we're moving away from the selectivity filter into something that's called voltage sensing domain. So it's a part of the protein that senses the change in the voltage across the membrane and triggers a motion, which typically leads to opening or closing the channel, right? That's the gating mechanism. And in the literature, many mechanisms have been proposed. None of them has been directly observed, because it's hard to observe that directly. It's also hard to do dynamics. It's hard to study dynamics with many established structural biological tools. Like, you cannot do dynamics in X-ray, right? That's what I mean. But our colleagues were able to design this experimental cell in which they put the gold layer here. They attach the membrane lipid bilayer to the gold. They put the protein, and then they can change the voltage here. And in this preliminary study here, they show that if they put a small peptide, it initially positions itself like this. This is alpha helix, this is a 310 helix. There are two different secondary structures. At 0 voltage, then what happens is they increase the voltage, it goes deeper in the bilayer, and it changes the angle between the two, and an extent of the two secondary structures changes by a little bit. So they were able to observe, this is the change in the line shape from 0 to 900 V. So there's a clear difference. This peak become more intense, right? And then this peak corresponds to or increasing the fraction of the one of the secondary structure, and they were also able to see that one of them, like that the peptide goes deeper in the bilayer and the angle changes. So now we're applying these ideas to voltage sensing domain of proteins. Hopefully we can depict, or we can see one of the mechanisms in actual motion, because you can resolve it with 2D-IR. It's fast, right? So you can resolve this motion. All right, this is my acknowledgement slide. I mentioned this already. So thanks to my collaborators, my group members, and thank you for your attention.
All right, let's thank Alexei. Do we have questions? Okay, yes.
Yeah, thanks. When you actually did the simulations, did you just simulate the selectivity filter itself, or any of the larger protein scaffolding holding it?
We simulate. Have an actual, goes all the way back here. So this is a snapshot from the simulation. Selectivity filter is only this part. So we've simulated the pore domain, all four units of the pore domain together.
What I was curious about is to what extent the protein fluctuations play a role in the dynamics of the ions in the water. So if you can imagine holding the selectivity filter rigid and saying, okay, do your stuff, water, or do your stuff, potassium. But now if you put it in the context of a fluctuating biomolecule, is that the same physics?
So selectivity filter is actually not rigid at all. And of course, in the simulations, we didn't keep it rigid. So yeah, it's actually one of the things that I skip. Very early, going back to 70s, a snag feed mechanism was proposed, and the idea is that so the selectivity field is just so perfectly built for potassium, so it's like a snag feed for them, and sodium doesn't find itself in such a nice environment as potassium inside. But then simulations show that selectivity filter can adapt. So if you put sodium, it will adapt and create a similar environment for sodium. So it's not rigid, and the simulations did not force it to be rigid. Yeah. So it does affect dynamics. It does adapt to an ion.
I guess one of the conclusions you had was you were saying now you have evidence that water is there, but basically in the case where the molecule the protein channel is closed, right? So do you think there could be a change, like when it basically is open, as to whether or not water would be there?
I think it has to do more with the conditions of the experiment, in the sense. So the experiments were done back in the day at 0 voltage. Now they can do it non-zero voltage. So in the literature, it looks like if you do, at least in the simulation literature, right, at 0 voltage you have a slight preference for hard knock, but at non-zero voltage you have a huge preference for hard knock. So I think the conclusion of this work might change if we do experiments that would be a different work, right? So if we do experiments at non-zero voltage, we might see more of hard knock. Now, to answer your question, it probably doesn't really matter what's going on here, because the ions are still there, so probably won't change much. What would change much is if we apply non-zero voltage.
Yeah. Okay, that makes sense. Do we have other questions? Yeah.
You had one slide where you listed like a bunch of different solvents, and then you had like the different frequencies in the Stark effect. But I was just looking at the list, and I know you said at the beginning of this talk that this region of the complex is like 4 angstroms wide, but like a bunch of those solvents are. Hexane's not going to fit through there, right? So like, if you're, I know it's not biologically relevant, but like you would have to at some point here, you'll have to go only into the hard knock mechanism, right?
Well, so this was just meant to illustrate that it's very sensitive to the local environment, and of course these environment inside are much more complex than just the solvent environment. Yeah, but it's just my message here was that the carbonyls are perfect or near perfect sort of reporters on the local environment.
Okay.
Yeah, so we're not putting those solvents except for water in there.
Can you give a flavor of like how long does it take to take these spectra? Like this is like huge averaging, right? That they have to do.
So the actual collection of the spectra takes minutes.
Okay.
Yeah. Now, because of the 25 years ago when this technique was developed, it was very slow. But now, with the pulse shapers and recent advancement, it's minutes.
Mm-hmm.
Yeah. So it takes the whole experiment will probably take a year, because you need to synthesize protein, ship it, and all that. But the actual measurement of 2DI it takes minutes.
Mm-hmm.
It's even averaging, decent averaging, it's still minutes. Yeah.
All right. And can you comment in your MD simulations, like what sort of force fields are you using, or like
So in MD simulation, I don't know. Let me show you this. So in MD simulations, we use. So there's this big debate in the community that one should use polarizable force fields for ion channels, because non-polarizable force fields do not handle ion carbonyl interaction correctly. So that's what we did first. We used polarizable force field to generate initial configurations. We used Drude. That's charm Drude. So it's developed by Benoit Roux. So we use polarizable force field to get this initial configuration. But then, because this map was developed for a non-polarizable force field, at the very last step we switched to non-polarizable and run a very short trajectory enough to calculate these frequencies. So this is about 20 picosecond trajectory that we used with the non-polarizable Gromos force field to calculate these frequencies. But the initial sampling was done with Drude, and it was probably 100 nanosecond or something like that for each configuration.
Yes.
It's a really naive question, but how realistic is this sort of the theorist's model that outside of the channel is water? You know, in an actual cellular environment, that's not literally what you have, but for the purposes of ion transport, is it a good model?
I would say what's important is, it is a very good question. So, very good question. So here on top, it's sufficient to have water and ions to answer your question. But there are some parts of this protein that are believed to be related to inactivation. So it's important to keep the entire protein for realism, basically, of the simulation. But it's enough to have water and ions, yes. In the sense that for this protein, it has not been reported that something else can go through or may impede or prevent transport. So essentially, N-type inactivation happens from here, but it's very slow, and we know it's the channel is closed, so you cannot inactivate closed channel, and nothing is happening on top of this. So,
I guess let's thank Alexei again. And anyone who has still questions, please come meet Alexei at the front here, okay? Thank you so much. Very nice.
thank you.
Very good talk.
I'll just leave it here. Where's the IT guy?
You'll pop up at some point, I guess. Self-driven. Yeah.
Did a really nice job explaining multidimensional spectroscopy. It's really hard to explain that to an audience. So
Yeah.
I tried, yeah.
What the heck are these double-sided Feynman diagrams?
Yeah,
Well, I didn't explain those.
Well, that was a good choice too.
Yeah.
But I meant to say, why is this ever? I need 4 pulses, right? You know, so what physically are you extracting from it?
Yeah, that's always a challenge. 2 weeks ago, I gave a talk at UC Davis, and that was Department of Physiology and I think you call it Department of Biology, Human Physiology, whatever. They actually have a pretty large crowd of ion channel people, and I gave a slightly different talk in the sense that I did not show some of the Feynman diagrams, but I still explained them to the So I had that slide with 4 pulses, but not the follow-up slide with the optical response function. Yeah. So there I have to explain the macroscopic polarization, and then briefly mention Maxwell equation, so it creates the field that's detected. So I had to really do it step by step.
Yeah, that's really.
Here I thought I don't need to go into those details.
Yeah, good.
that was a good choice, yeah.
That was nice. I enjoyed the talk. It was nice meeting you.
Right. Thank you, yeah. Good, yeah. Good. Okay, perfect. Yeah, nice.
Yeah, thanks, yeah.
Yeah, we don't have, we actually, some of the faculty we have, or have been working on ion channel, like nanopores, solid state nanopores, more than biological. So they weren't here, but I guess everyone's busy, so whatever. But yeah, no, I think it was really fun. Yeah, in this community, like some of the work I do is also like solid state channels. And like very often in that community, like nanophysics, people are showing the potassium channel and some of this, and they always say this is inspiration. We want to build that in like solid state systems and so on, right?
Yeah,
yeah. Someone doesn't know. It's like important.
Yeah. Fucking A, bro. So tired of it anymore. This is insane. Remember, it felt like they were right there.
I didn't even really hear him that well.
Really?
Yeah.
Yo, what?
Well, that's definitely one of ours.
This is one of ours, dude.
Yeah.
What is it?
A2000 Wilson?
Yeah. Dude, holy shit. Do you remember me going in here and checking to see if there's enough space in here? I was like,
Yeah,
fuck.
someone's inhaled or something there.
Yeah, me and
I was sitting in that corner.
Yeah.